Hello everyone, my name is Snehlata and I'm a PhD student and a PMRF fellow in the Department of Biological Sciences and Bioengineering, IIT Kanpur. Today we will continue with the next lecture of the chapter Biomolecules. So till now what we have studied about the classification of amino acids, and we saw that there are 20 standard amino acids, and these amino acids are classified on the basis of various criteria, such as on the basis of shape, structure, and on the basis of their requirement in the diet, etc. Now we will see how these amino acids are synthesized. So most of the amino acids are synthesized. Most of amino acids are synthesized by a type of acid that is known as alpha keto acid. Okay, so most of the amino acids are synthesized by this alpha keto acids. Now, what are alpha keto acids? So, as the name suggests, these acids have both keto group and acid group attached to their alpha carbon. For example, here we are seeing the example of two alpha keto acids. The first one is known as pyruvate. The first structure is pyruvate. Here, if we see, we can see that this first C, on which the functional group is attached, this carbon is known as alpha carbon, right? And it has both the ketone group, that is CO, and the carboxylic group, that is COOH. And it is a type of alpha keto acid. Similarly, we have a second, this is the first alpha keto acid. We have a second type of alpha keto acid, in which we can see that on the alpha carbon we have COHO, along with we have another carboxylic group attached after this ethyl group, right? Now, what happens? How does the amino acids are formed by this alpha keto acids? We can understand by the example. Suppose in this second structure, this structure is known as alpha keto glutarate, alpha keto glutarate. In this structure, if an NH2 group is substituted or it's replaced this CO group, then what do we have? We will find a structure that we have seen previously. So we will find a structure something like this, right? Here it will have NH2 to complete its valency, H will be attached and COH. So we can see we have seen this structure previously in our last class. What do we have here? It is an acidic amino acid. Right? What acidic amino acid? It is known as glutamate. This amino acid is known as glutamate. It has two C. This is the main region or main structure of all the amino acid that is common in all the amino acid. Then we have this R group. This is the R group, right? This is the R group that has CH2, COH, and this structure is known as glutamate. So by this example, we can see that how amino acid is formed by the alpha keto acid. So, and not only glutamate, there are most of the amino acids are synthesized by different type of alpha keto acids. Now, one more important thing, this glutamate is the first amino acid to be synthesized. Okay, so this is the first amino acid that is synthesized in a cell, and after this, by using the substitution reaction and various other reactions, different other amino acids are synthesized. Second important thing is that amino acids are also called as alpha amino acids. So why do we call them as alpha amino acid? The answer is simple. So if we see this figure, this structure, this is the alpha carbon, and on which the amino group is attached, right? And because of this reason, the amino acids are also called as alpha amino acids. So we can write, because the amino group we have, the main amino group is attached to alpha carbon, right? And because of this reason, Because of this reason, the all amino acids are called as alpha amino acids. So this is an overview of this biosynthesis of amino acid. The important part is that most of the amino acids are synthesized by alpha keto acids. Now we will see what is the role of amino acids. So the first role that we all know and that we are studying is it is a building block of protein, right? So the first role is it is the building block of proteins. So proteins are synthesized by amino acid, right? Now, second thing, there are various hormones such as thyroxine. What is thyroxine? These are T3 and T4 hormones. Then we have other hormones like dopamine, the happy hormone, adrenaline, and melanin. So these hormones are synthesized or are derived by a amino acid that is tyrosine. So these are derived from tyrosine. Okay. Now, similarly, we have auxin, vitamin B12. These components are derived from tryptophan. Okay, tryptophan. And what is tryptophan? Tryptophan is most complex amino acid, right? It is most complex amino acid. Now, if we talk about artificial sweetener, okay, so the artificial sweetener, so it is derived from an amino acid that is aspartate. And aspartate is acidic amino acid, right? There are two acidic amino acids, aspartate and glutamate. So the artificial sweetener is derived from aspartate. If we talk about histamine, then these are derived from another amino acid that is histidine. And what are histidine? Histidine is basic or positively charged amino acid, right? Basic amino acid. Okay. Now, so we have seen there are several other various roles of amino acid, but these are the roles that are very important. Apart from this, if someone asks which is the simplest amino acid, so we have seen the simplest amino acid is glycine, right? Okay. So with this, we have completed about the amino acids, what are different types of amino acids, their classification, and how amino acids are synthesized. Now, what we will see, now we will first solve a few questions, then we will move to the next part of this chapter. So the first question is, the simplest amino acid is, so I just told you the answer is glycine, right? Now, the second question is, amino acids are mostly synthesized from, so they are synthesized from alpha ketoglutarate acid that I just told you, okay? The third question is, the amino acids with the aliphatic R group are. So there are various options. So I gave one acronym to remember this. That was all vans are interesting, right? And then I also saw that there is one simple amino acid, so you can add it here. Okay. So if we go by this thing, what is the first amino acid? That is glycine. A stands for alanine. V stands for valine. Sorry, this is. All vans is. Sorry, one second. Okay, so it was, the acronym was all vans look interesting. Sorry. So the acronym was all vans look interesting. Okay. So here we look. Okay. So L stands for leucine and I stands for isoleucine. So with this, if you check the options, we have first option glycine, alanine, and leucine, right? So glycine, alanine, and leucine. So these are the aliphatic amino acids that contain aliphatic R group. Now, the next question is, which amino acid is not important to be taken in diet? So the amino acids that are not important to be taken in diet are known as non-essential amino acids, right? They are called as non-essential amino acids. Now, what were non-essential and essential amino acids? So, if you remember, the non-essential amino acids, for the essential amino acids, we had one acronym, and that was I love my hairy little tiger very truly and passionately, right? And passionately. So if you go back to the lecture, you will see that what these each amino acids, the first letter of these represent one amino acid. Now, if you go by option, we have histidine here, and we can see we have H, right? So histidine cannot be the right answer. The second is threonine. We have T that represents the threonine. The threonine cannot be the right answer. Then we have serine. In this acronym, we do not have S anywhere. So the correct answer is serine, right? This is a non-essential amino acid. And this acronym is for essential amino acids, right? So the remaining are non-essential. So with the correct answer here is serine. This is a non-essential amino acid. Apart from this, all other amino acids are essential. Okay. So now the next question is, the naturally occurring protein consists of. So what we saw that the naturally occurring proteins are made up of L amino acids, right? We had two type of amino acids based on their ability to rotate the plane polarized light. So if the light is in a particular plane and if it is passed through a mixture, then if the light is rotated left or right, this mixture rotates the light in the left direction, this is known as levorotatory. The mixture is known as levorotatory or L, right? And all the naturally occurring protein consists of this L amino acids, because these amino acids rotate the plane polarized light into the left direction. Okay. Okay. So with this, we have completed our amino acid part. Now, the next thing that we will see is the proteins. So, what are proteins? Proteins are nothing but they are chain of amino acids, right? These are chain of amino acids. This thing we have understood till now. Now, what are the typical features of proteins? So, the first feature of protein is it is biomacromolecule. Proteins are biomacromolecules. So if you remember your first lecture, so what I told about biomacromolecules, these are the molecules which have a molecular weight. So these are the molecules which have molecular weight greater than 8,000 Dalton, right? So the proteins have molecular weight greater than 8,000 Dalton. Now, the second part about biomacromolecules is that if you crush a tissue or a cell or a leaf and then you dissolve it into an acid that is trichloroacetic acid and then try to dissolve its component by passing through it through a filter, then we saw that we will obtain two fractions. One was the retentate that will be retained in the funnel, and the second was filtrate that will pass through the filter, right? So what we saw in that case, that the biomacromolecules, because they are of large size, they cannot be passed through the filter. So they will come in what pool? They will come in either, you can say, acid insoluble pool, because they are large in size, so they were not dissolved, or they will not be dissolved in the trichloroacetic acid. So they will become under acid insoluble pool. Or you can say, if you pass it through the funnel, it will be in the retentate part, right? So the proteins come under acid insoluble pool or the retentate part. Now the third thing is, the proteins are heteropolymers. Proteins are what? These are heteropolymers. Now, what do I mean by heteropolymer? Hetero means different, right? So they have different monomeric units. So if I'm saying that a protein is a chain of amino acid, by this I mean that it has glycine, alanine, threonine, methionine, right, isoleucine, So this chain is looks something like this. It will never look like this GGG, that it has a chain of glycine only or it has a chain of alanine only. So it will not look like this. Okay. So it will always be heteropolymer because it will contain different monomeric unit. And by monomeric unit, what I mean, that different amino acids, because the monomer of protein is amino acid, right? So it will have different monomeric unit and hence it is a heteropolymer. Now, if we talk about the nature, of amino acid, so what is the nature of amino acid? It is amphoteric. The nature of amino acid is amphoteric, which means it can act both as acid and base. Okay. Why? Because if you remember, the amino acids are zwitterion, right? So based on the pH of the solution, they can either exist as a positively charged or negatively charged, which means they can either give their proton or hydrogen ion or they can accept the hydrogen. And because of this feature, they can act as both acid and base. And the solution or the compound that can act as both as acid and base is known as amphoteric. Okay. Now, if we talk about the abundance of the protein, so in a cell, if we talk about the abundance, so it is the second most abundant component, or you can say biomolecule in the cell. Okay. So in cell, proteins are approximately 10 to 15%. Okay. So 10 to 15% of the cell consists of protein, and it is second most abundant biomolecule. Now, which is the first most abundant biomolecule, or you can say molecule? It is water. Okay. So water is the first abundant molecule that is present in the cell. That is 70 to 90%. Okay. So this is water is not a biomolecule. Okay. In the first lecture, I told you the definition of biomolecule. So according to that, water is not a biomolecule. So the most abundant biomolecule is protein. Okay. But if someone asks you which is the most abundant molecule, then you will have to answer water. Now, apart from this, there are two more things. So if someone asks you that most abundant protein in a cell, okay. So the most abundant protein in cell. So in cell, the most abundant protein is collagen. Okay. It is the most abundant protein. And by cell, I mean animal cell. Okay. In animal cell, the most abundant protein is collagen. But the most abundant protein in entire biosphere, if someone asks the most abundant protein in entire biosphere, so the answer is rubisco. So rubisco is an enzyme that is used in the dark reaction of photosynthesis that we will see later. So the most abundant protein in entire biosphere is rubisco, but in animal cell, the most abundant protein is collagen. Okay, so these are the basic features of the protein. Now let's talk about the difference between a protein and a polypeptide. Okay, so what I said, that protein is a chain of amino acid. If someone asks you what is polypeptide, the answer will be same. It is a chain of amino acid. But before moving to that, first let's see how a protein is formed from an amino acid. Okay, so for that, let's draw one amino acid. So we know on the amino acid we have COH group, we have NH2 group, right? We have NH2 and we have one from R group. So suppose this R group is R1, it is representing any amino acid. Now, let's draw another amino acid. So we have NH2 group here again. This is NH2. We have C alpha. We have COH group attached. We have hydrogen. And we have, say, another functional group R2. That is representing a second amino acid. Now, how does a protein is formed? So a protein is formed by when the NH2 group of one amino acid react with the carboxylic group of another, and it causes the release of water molecule. So water molecule is released and it results in a structure that will look something like this. So it will result in something like this, right? It will have CO group, it will have NH group, right? And it will have C alpha, COH, hydrogen, and this R2. Now, this bond that is formed, this bond is known as peptide bond. This bond is known as peptide bond. Okay. Now, and this structure, because it comprises of only two amino acids, right? So this structure is known as dipeptide. Okay. So this structure is known as dipeptide. Why? Because it comprises of only it is formed by reaction between two amino acids. Hence it is called dipeptide. Similarly, we have tripeptide that is formed by three amino acids. Okay. Now, if this chain is formed by multiple amino acids, that is more than say 10 amino acids, then the structure will be called as polypeptide. So what is polypeptide? So the structure polypeptide, when these multiple amino acids are linked to each other via this peptide bond formation, then this chain will be known as polypeptide. Okay. So what we saw, and this chain is linear, right? So we can write that polypeptide is linear chain of amino acid. It is linear chain of amino acid that is formed by joining multiple amino acids via peptide, right? Now, when this polypeptide is folded, okay, this polypeptide, when it gets folded, okay, it folds, it becomes functional. It becomes biologically functional. Then this structure is known as protein. Okay. So this is the difference between protein and polypeptide. Polypeptide is a linear chain of amino acid. While if you talk about a protein becomes when a polypeptide chain folds and it becomes functionally or biologically functional. Then this structure is known as protein. Okay. So this is the difference between polypeptide and protein. Now let's talk briefly about the peptide. So let's talk about the peptide bond. So what is peptide bond? Peptide bond is nothing but it is formed by reaction between amino group of one amino acid and carboxylic group of second amino acid, right? So this is the peptide, right? Now, if we talk about the feature of peptide bond, the peptide bond it is also known as amide bond. Okay, it is also known as amide bond. Secondly, its nature is it is a covalent bond. Okay, it is a covalent bond. It is forming by the sharing of electrons, right? So it is a covalent bond. Okay, so this is about peptide bond, and now we have seen the difference between protein and polypeptide. The folded form of a polypeptide that is biologically functional is known as protein. Okay. Now, but does it mean that all the polypeptides are non-functional? No. The answer is some of the polypeptides are also functional. Okay. So they are also biologically functional. And one of such example is growth hormone. So certain polypeptides that are linear chain are also functional, and the example is growth hormones. Okay. Now let's talk about the structure of proteins. So the structure of protein here, what we are seeing the linear chain of amino acids that are joined to each other. This is the first amino acid, it has R1, then this R3, then there is R2, right? So this is how it is represented. This is R3. So these are three amino acids that are joined to each other. And where do we have peptide bond? The peptide bond is formed between this CO and then this NH. Okay, so this is the peptide bond, right? Now, we have one more thing. We have, if we look at the first amino acid or the amino acid that is present at the left corner, then this left amino acid have a terminus, okay, or have amino group, free amino group attached to it, which is known as left amino acid, this amino group, which gives this complete protein a terminus that is known as N-terminus. So left amino acid has N-terminus or amino terminus. Okay. So in protein, we have two terminals. One is known as N-terminal and the second is known as C-terminal. By N-terminal, what I mean? What I mean is what group is present at the leftmost or the first amino acid. So left amino acid or at the first amino acid, what group is present? So at the left amino acid, we have NH2 group present, right? This NH2 group is present, and it gives it as, and because of this reason, it is called as amino terminus or N-terminus. Similarly, if we talk about the right amino acid, the rightmost amino acid or last amino acid. So on the rightmost amino acid or the last amino acid, what group we have? We have this COOH group, right? This carboxylic. We have the carboxylic group. And because of this reason, this terminal is known as C-terminal or carboxylic terminal. So the right end of chain is known as carboxylic terminal, is known as carboxylic terminal or C-terminal. We understood, right? Because whatever the length of the protein we have, but always at its leftmost end, it will have amino terminal because it will have this amino group free, and at the rightmost terminal, it will have carboxylic. The terminal is known as carboxylic terminal or C-terminal because its carboxylic group is free. Okay, so the amino acids, in simple terms, you can write protein chain have two terminals. Have two terminals, and what are these? Amino terminal, amino or N-terminal, and second is carboxylic or C-terminal. And we know why these are known as amino terminal and carboxylic terminal. Now, if you look at the structure of the protein, so there are four layers of structure in a protein. The first layer is known as primary layer. Okay, so the first layer is known as primary layer. The second is known as the secondary layer. The third is tertiary layer, and the fourth is quaternary layer. So it has four layers, or you can say four types of structure. Protein has four types of structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Okay. Now let's study these structures one by one. So the first structure is primary structure. So what is primary structure? The primary structure is the basic structure. It is the basic structure of protein. Okay, this is the basic structure of protein, and it is also known as 1 degree structure. Also known as 1 degree structure. Okay, it is known as 1 degree structure. Now, what do we have in the primary structure? So if you look at the figure in the primary structure, here we can see these are the chain of amino acids, right? They are one amino acid, second, third, fourth. So these are the chain of amino acids which are linearly arranged. So these are, in the primary structure, we have linear chain of amino acids, and we know that these amino acids are connected by peptide bonds, right? So they are joined by peptide bonds. Okay, so in the primary structure, what bond we have? We have only peptide bonds. Now, if we talk about what kind of information this primary structure gives, so it gives two types of information. The primary structure gives information. It gives information about, it gives information about, first thing, what all amino acids are present in that protein, right? So it gives us information about the sequence, about the sequence. By sequence, what I mean that what amino acids are present, what all amino acids are present in that chain, right? So, or we can say that what amino acid, this protein is rich in what kind of amino acid. So all this information we can get from the primary structure. The second information it gives is about positional information. It gives information about positional information. It gives positional information. What do I mean by positional information? It means that order of amino acids. So order of amino acid, that whether that if someone asks that which amino acid is present at the 4th position, which amino acid is present at the 10th position, then by analyzing the primary structure, you can give this information, or you can get to know about this information. So the primary structure gives the information about the sequence and about the positional information. Okay. Now, next question is, how does the amino acids know that in what order or in which order they have to be arranged? So this information, from where they get this information? So this information about the arrangement, the information about the arrangement of amino acids in protein is stored. It is stored somewhere. Okay, this information is stored somewhere, and it is stored in DNA. Okay, so this information about the order and what all amino acids will be present in a protein is stored in DNA. Okay, so this information is coded by DNA. Okay, right. So this is all about the primary structure. Now, next we will move on to the secondary structure. Okay, so in the secondary structure, when does the secondary structure forms? So the secondary structure is formed when the primary structure, is formed when the primary structure folds and twists upon itself. Okay, folds and twists upon itself. So when it folds and twists upon itself, it gives the secondary structure, or this is also known as 2-degree structure. The structure is also known as 2-degree structure. Okay, so the primary structure, that is a linear chain, it folds and twists upon itself to give the secondary structure. Now, the secondary structures are of two types based on how this protein or the primary structure is being folded. Okay, so there are two types of secondary structure. The first one is the alpha helix. Okay, so the alpha helix looks something like this. Okay, so it is looking something like this. So what we can see, it is a helical structure, like a spring we have seen, right? Or a rope, you can say. So it is a helical structure. Now this helical structure has certain axis, right, along which it will be rotated. For example, if you take a pen and then you start rotating start rounding or wrapping a thread over that pen, you will get a structure that is known as helical structure. Now, in that structure, what will be your axis? The axis will be that pen on which you are wrapping that thread, right? So similarly, the protein has certain axis along which the amino acids or the chain of this amino acid is rotated or is wrapped around. Okay, so this is that axis. Now, if we talk about the bonds, so it has certain axis. This is the axis, right? Now, the helical structure, these helical structures are always right-handed. Okay, so the structure, if you talk, so it will always move through this clockwise direction on that thread. On that pen. So what I'm trying to say is, if you take a pen and then you start wrapping a thread around it, then in the case of alpha helix, what happens? The thread will always be rounded in a clockwise direction. Okay, it will always be in the clockwise direction, and because of this is the alpha helix. So if someone asks you about the nature of the alpha helix, so what you can write, that the alpha helix is in protein. In protein, alpha helix are always right-handed. Okay, they are always right-handed. Right-handed means they are rotating clock in the clockwise direction. Okay, so they will be rotated in the clockwise direction along this axis. Okay, now what are the bonds that are present in the alpha helix? So bonds. So what we have seen that this is the primary structure that is being folded, right? So the bond that is present in the primary structure will also be present here. So the first bond that will be present in the alpha helix is peptide bond, right? Because peptide bond is present in the primary structure. But apart from peptide bond, this alpha helix is also stabilized by another type of bonding. Okay, so suppose here is one amino acid and here is second amino acid. So there will be certain interaction between these amino acids, and this interaction is known as these amino acids interact by a hydrogen bonding. So the second bond that is present in alpha helix is hydrogen bond. Okay. Now, this hydrogen bond, if you talk about the direction of the hydrogen bond, what we can see that this is the hydrogen bond, right? This is the hydrogen bond, and this is the axis. This is the axis of that alpha helix. So what we can see that this hydrogen bond is perpendicular to this alpha helix. So direction of hydrogen bond is parallel. It is parallel, right? It is parallel. So it is parallel to axis of alpha helix, right? It is parallel to the axis of alpha helix. Now, if we talk about how many amino acids are present per turn, so this is one turn, right? From here, if you say, so this is one whole turn. This is one whole turn, right? So how many amino acids are present from here to here? So the amino acids per turn in alpha helix are amino acids per turn in alpha helix are 3.6. Okay, so there are 3.6 amino acids per turn in alpha helix. And if we talk about the one example of a protein that exists in this alpha helix conformation, then the example is alpha keratin that is present in our hair, right? So alpha keratin is one of the example of alpha helix. Now, the second type of secondary structure is beta sheet. Now, what is beta sheet? So beta sheet looks something like this. Okay, so it will look something like this. Now, so it will look like it looks like a sheet-like structure. It looks like sheet. So it has sheet-like structure. This beta sheet is also known as beta pleated sheet. Okay, so if someone has beta sheet or beta pleated sheet, both are same. Okay, now again, what bonds are present in the beta sheet? The same as the alpha helix. So it has peptide bond. The beta sheet has peptide bond because it is formed upon the folding of the primary structure. So it has peptide bond plus it also has hydrogen bond. Now, how hydrogen bonds are attached here? So hydrogen bonds will be formed something like this. So here is one amino acid, here is second amino acid. So hydrogen bonds are formed something like this. Okay. So the direction of hydrogen bond, and this is your axis. This is your axis, right? So the direction of hydrogen bond is perpendicular. It is perpendicular to sheet axis. Okay, it is perpendicular to sheet axis. And then if we talk about the example is fibroin. So it is a protein present in silk. It is present in silk. So it is in the beta sheet conformation. So based on the secondary structures, there are two types, alpha helix and beta sheet. And you can also see the same thing via this diagram. So the first diagram here is the alpha helix. Okay, it is alpha helix. And the second diagram is beta sheet. Okay, so this secondary structure also one more important thing about the secondary structure, it is of the only one polypeptide chain. Here, although you are seeing the beta sheet, but they must be connected somewhere. Okay, they must be connected somewhere. So it is a single polypeptide chain that is folded in such a way that it is forming two sheets. Okay, but it is a single polypeptide chain. It do not consist of multiple polypeptide chains. Okay, now the next structure is the tertiary structure, and this is also known as 3-degree structure. Okay, it is also known as 3-degree structure. So what is tertiary structure? Tertiary structure is formed further when the secondary structure further folds upon itself. Okay, further folds upon itself. It further folds upon itself to give a structure like a wool. So we have seen wool, right? So it folds upon itself to give structure like wool. Structure like wool. Okay, so this structure forms a tertiary structure. Now the tertiary structure is the 3-dimensional structure. It is the 3-dimensional structure of protein. Tertiary structure is the 3-dimensional structure of protein. Now, what all bonds are present in the tertiary structure? So bonds that are present in the tertiary structure. So the first thing, let's see, because it is formed by the folding of the secondary structure, and the secondary structure itself is formed by the folding of primary structure, right? So it will consist all the bonds that are present in the primary structure as well as the secondary structure. So let's write. So the bond that is present in the primary structure is peptide bond. It is peptide bond. Now, what bond was present in the secondary structure? That was hydrogen bond. Hydrogen bond. Now, apart from this, what other bonds are present in the tertiary structure? Let's see. So, your tertiary structure will be formed something like this. So, it will be formed by the folding of the secondary structure. So, it will have alpha helix, right? Then it will have sheet. Suppose again it will have helix, then sheet, then helix again, right? Then helix again, something like this. Then here is sheet, something like this. This protein structure tertiary structure will be formed. Now, if you see, and this protein, where it will be present in our body, it will have aqueous media or say water surrounding it. So it will have water, or in the cell also it will have water around it, right? So now, see, suppose here is one amino acid. The one amino acid is present here. Now this amino acid will interact with the water, right? It will interact with the water. It will interact with the neighboring amino acid that are present inside. Similarly, if there is one amino acid here and one amino acid here, and it is present in the core, that is, it is not exposed to the water, it is present inside. So these amino acids must be non-polar, right? Because non-polar amino acids are the one that do not interact with the water. So they form an aggregate kind of structure so that they don't get exposed to the water. But these amino acids, because they are of non-polar nature, have certain interaction with each other. And this interaction is known as hydrophobic interaction, right? And this interaction with the water is known as either it will form hydrogen bond or, it will form hydrogen bond. Similarly, suppose you have one charged amino acid here and another charged amino acid here. These two amino acids will interact with each other, right? And this interaction will be electrostatic interaction. So all the non-covalent interaction that we know will be in the tertiary structure, right? So it will have, along with the peptide bond, hydrogen bond, because of the non-polar amino acids, it will have hydrophobic interaction, or you can say hydrophobic bond. Hydrophobic bond. Then, if there are two charged amino acids, if they will interact with each other, then it will have ionic interaction. It will have ionic interaction. It will have van der Waals interaction. It will have van der Waals interaction, right? And suppose it has sulfur containing amino acid, that is cysteine, we know, right? So suppose it has cysteine here, this amino acid is cysteine, so it will have sulfur. Similarly, here also this amino acid is cysteine, it also will have sulfur. then they will form this disulfide interaction. So they will also have disulfide interaction, right? So these are all interactions that are present in the tertiary structure of a protein. Okay. Now, one more important thing about this tertiary structure is that this structure, if you look at the structure here, you can see this is forming a pocket-like structure. This will be a pocket where a substrate can bind, right? So the tertiary structure forms a binding pocket. It forms the binding pocket, or you can say this is also known as active site, that we will study in more detail when we will study enzymes. Okay, but for now, you remember that tertiary structure forms the binding pocket or active site. And one important feature of this tertiary structure is that it is of tertiary structure is of utmost importance. It is of utmost importance for biological functioning of protein. For biological functioning of protein. Okay, it is of utmost importance for the biological functioning of protein. So tertiary structure is important for the biological functioning of a protein because it is here, what you can see, it is forming a binding pocket or active site on which the substrate will bind and further biological function will take place, that we will see in detail. But for now, you remember that is very important for the biological functioning of protein. Now, we have studied about the tertiary structure. The next structure that we will study is quaternary structure. The quaternary structure is also known as 4-degree structure. And the protein is only molecule that go up to 4-degree structure. Okay. So, what we have in quaternary structure, the first important thing about quaternary structure is it is comprises of more than one polypeptide chain. Comprises of more than one polypeptide chain. Okay. So till now what we have seen, the 3 types of structure, primary, secondary and tertiary, they had only one polypeptide chain. That polypeptide chain was being folded upon each other and via various interaction, it was forming primary, secondary and tertiary structure. But the quaternary structure have more than one polypeptide chain. And these polypeptide chain, the single polypeptide chain is known as subunit. Okay. The single polypeptide chain is known as subunit. Okay. So it is known as subunit. A single polypeptide chain is known as subunit, and a quaternary structure is made up of more than one subunits. Okay. Now these subunits or these single polypeptide chains, they arranged. So subunits are arranged to form quaternary structure. Okay. They are arranged to form quaternary structure, right? And if we talk about the example, the first example we have is hemoglobin. So hemoglobin is oxygen carrier, right? It is present in our blood, RBC, right? And it is oxygen carrier. So hemoglobin, it has 4 subunits. Okay. Hemoglobin has 4 subunits. So what do I mean by 4 subunits? It will have one tertiary structure, something like this. Second is something like this. Third, okay, and fourth. So these 4 together are forming the structure of protein, that is hemoglobin. Now these 4 subunits, they are further named. The 2 of these subunits are called as alpha subunits. So it has 2 alpha subunits and other 2 plus 2 beta subunits. Okay, so they have 2 alpha subunits. Say these 2 are alpha subunits and these 2 are beta subunits. It comprises of 2 alpha subunits and 2 beta subunits. The second example is, the first example is hemoglobin. The second example is insulin. So insulin, okay. Insulin is a hormone, okay, that regulates the blood glucose level. Okay, so this insulin also exists in quaternary structure. But what is difference between the structure of insulin and hemoglobin is that insulin, one of the differences is insulin, it has 2 subunits. The hemoglobin has 4 subunits, insulin has 2 subunits. And the name of these subunits are alpha and beta. So these are alpha and beta subunits. The insulin have alpha and beta only one alpha and one beta subunits, while the hemoglobin has two alpha and two beta subunits. And one more important thing is it does not mean that the alpha subunit of hemoglobin and alpha subunit of insulin is same. They can be different. It's just a name. Okay. So with this, we have completed the structure of a protein. So we saw that there are four types of structure: primary, secondary, tertiary, and quaternary. And in quaternary structure, there is more than one polypeptide chain, but in rest of three structures, it has only one polypeptide chain. Okay. Now, next is there are types of protein that is based on the shape. So we'll talk about the types of protein, and it is on the basis of shape. So the first type of protein is fibrous protein. So what is fibrous protein? Fibrous protein, so if you look at the structure of the fibrous protein, they will look elongated. So they have elongated structure. They have elongated structure. Okay, they have elongated structure. Second thing, these proteins are not soluble in water. Okay, they are not soluble in water. You can say water insoluble. They are water insoluble. So third thing, if you look at the structure of this fibrous protein, they will be mainly either in the primary structure or secondary structure. They have little or no tertiary structure. So a very small amount of fibrous protein can have tertiary structure, or no, they do not have tertiary structure at all. So little or no tertiary structure. Okay, it means that they mainly exist in primary and secondary structure. Okay, they mainly exist in primary and secondary structure. Now, if we talk about the function, what is the function of fibrous protein? So they are mainly involved in structural support. They involve in structural support. Okay, and what are the examples of fibrous protein? The examples are collagen. Collagen is one of the most abundant protein that is present in the cell. Then we have other proteins like actin, myosin that are present in muscles. Then we have a protein that is present in our hair, that is keratin. Okay, so this is about the fibrous protein. That is a type of protein based on the shape. So it has an elongated structure. Now the second type of protein is globular protein. So the globular protein, they have what we saw in the case of fibrous protein, they have little or no tertiary structure, right? They mainly exist in primary or secondary form. But the globular protein, they mostly exist in tertiary structure. So exist in tertiary structure. And this tertiary structure, it gives it this globular spherical shape. It gives it globular or spherical shape, right? And there are certain other globular proteins that exist in quaternary structure. So they mostly exist in tertiary structure, but some of them exist in quaternary structure as well. Some of them exist in quaternary structure. Okay, then these proteins are soluble in water. These are soluble in water. Okay, and their main role, like the fibrous protein, they has main role in structural support. This protein have main role in metabolic reactions. So they have role in metabolic. They have role in metabolic reactions. One of the example is enzymes. So enzymes are proteins. Okay, most of the enzymes are proteins, and these are globular in nature. Okay, if you look at the structure, so here the A structure is, this is what? This is fibrous protein. So we can see they have elongated structure, right? And this is the second is, this is globular protein. So they are folded in a such a way that they have this tertiary structure that is spherical, looking like spherical in shape. Okay, so these are the structures of this fibrous and globular proteins. Now the next thing, is, let's talk about the function of some protein that is given in the NCERT. So the first function, the first protein that is given is collagen. So the first protein is collagen. So the collagen, I just told that the collagen has role in structural support. This protein has role in structural support. And here what is given, that it is intercellular ground substance. So intercellular means between the cells. Okay. So suppose you have one cell, you have two cells, right? So between these cells, how these cells are attached? These are attached where this collagen. There are other proteins are also present, but mostly it comprises of collagen. So it will have collagen in between. Okay. So hence their function is intercellular ground support, and which in turn is providing the structural support to these cells. The second protein is trypsin. So trypsin is an enzyme, and trypsin is an enzyme, and this enzyme is digestive enzyme. Okay, so it helps in digestion. And the enzymes are, what are enzymes? Not only trypsin, most of the enzymes are proteinaceous in nature. Okay. Now the third protein is insulin. So what is insulin? Insulin is a hormone, and what it does, it regulates blood glucose level. It regulates blood glucose level. Okay, it regulates blood glucose level. Then the fourth is antibody. So antibodies are what? These are the molecules that are produced by immune cells, right? So that they fight against infectious agents. And one of the example of antibody is IgG. Okay, IgG. It is globin protein. Okay. Now the fifth is receptors. So what are receptors? Receptors are, suppose this is a cell. So on the cell membrane, these certain protein molecules are present on which certain molecules bind, and that leads to the reaction inside the cell. Okay, that thing we'll see later. But for now, just remember that these are the molecules that are present onto the cell membrane or nuclear membrane, and in some cases they are also present in the cytoplasm. Okay. Now the sixth is GLUT4. So what is GLUT4? The GLUT4 is, suppose this is your cell, this is your plasma membrane, then the GLUT4 will be present over the plasma membrane. It is a channel. Okay, so GLUT4 is a channel, and what this channel do? This channel allow transport of glucose. It allows transport of glucose. Okay, so what is GLUT4? GLUT4 is a channel that allows transport of glucose. So these are certain proteins and their functions. Okay. Now, okay. So after this, let's talk about one topic, that is denaturation of protein. So what do we mean by denaturation of protein? So denaturation means, it means disturbance of, denaturation means disturbance of shape and stability of protein. Okay, disturbance of shape and stability of protein. Okay, so now what I mean by this, so we know that a protein exists at most, a biologically functional protein exists in the tertiary form and also in the quaternary form, right? So let's draw a structure of a protein. So this is, suppose, a tertiary structure of a protein. Okay, it is tertiary structure of protein. So it is forming like this. Now, because of its folding, suppose it forms a binding pocket here. It forms a binding pocket here, and due to which it performs a particular function. Because in this binding pocket, a substrate will bind, and then a certain reactions will happen that will allow the metabolic functioning of the cell. Okay. Now, suppose this protein is kept in an environment that changed. Okay, that environment in which the protein is kept is changed. So if there is change in environment, if there is change in the environment of the protein, and by the change in environment, what I mean, suppose there is change in the temperature. So the where the protein is present, suppose there is change in temperature, suppose there is change in pH, suppose there is change in the solvent, or the solution that is around this protein. So it might be possible, or in some cases what happens, this protein comes in its native structure. So it forms a structure something like this, a linear structure. And this, you can say, this forms its native structure. And what is the native structure of the protein? It is the primary structure, right? It is the primary structure. And this primary structure, what it has? It has a peptide bonds. The primary structure has peptide bonds. Okay. So this peptide, what happened? Now the protein that was in the tertiary structure was able to perform a certain function, but now due to the change in the environment, such as temperature, pH, and solvent, it comes under this native structure. So it won't be able to perform its function, right? So what it will lead to? It will lead to the disruption in the function. First, the shape was changed, disruption in shape and function, right? It leads to the disruption in shape and function. And this state is known as denatured protein. This is known as denatured. This is known as denatured protein. Okay. So this is what I mean by denaturation of protein. And the denaturation of protein happens by the change in environment such as temperature, pH, and solvent. So with this, we have completed our topic, that is protein. Now in the next lecture, we'll study about enzymes. That's all for now. Thank you.